So I thought it would be a good idea to start off reviewing an important concept that we established in our last lecture. Alright, so let's look at this video, and I pose a question. What type of mobilization is the fly practicing? So in our previous lecture, we talked about this concept of organisms taking advantage of the environment, specifically taking advantage of essential metals, and they do so by mobilization. And so we talked about a few methods of mobilization, and so what this fly is doing is one of them. Alright, so for a little bit of context, what the fly has done, it has vomited its digestive enzymes, which is actually quite acidic. So essentially, what the fly is using is an acidification process to degrade these components so that it can be able to obtain its nutrients. All right, so we're essentially looking at acidification here. This lecture builds off of many of the themes that we discussed in our last lecture, which focused on the ideas of traveling through membranes. And so here we're going to focus on the specific transports and trafficking that relates to iron in mammals. Here were the highlights of our previous lecture. We focused on the basic composition of biological membranes and emphasized the requirements for solute membrane permeability. In this discussion, we focus on the distinction between passive versus active transport. And then we focus our discussion on the concept of metal mobilization. And so we emphasized three key things. That is chelation, which was the focus of the remainder of the discussion. We also talked about redox processes and acidification, which is just the example that I showed you with the fly. Now let us draw our attention to iron. As we know, iron is an essential metal, and our body maintains a homeostasis of this metal in order to be able to survive. And so what we see here is a list, and I would say this is a minor list. If you think about the different types of iron-based proteins and how that metal is utilized either directly or indirectly to facilitate certain key functions or key molecular mechanisms. So let's look at some of these in this list, and in a way this is a review for what we've already talked about. We are all familiar with hemoglobin. So let me turn on my pointer. So we're familiar with this protein. It is a heme-based protein, and its function is to facilitate the binding of oxygen to the iron to enable transport of the nonpolar molecule. Myoglobin is part of this family of proteins, and its function is to facilitate storage of oxygen. We also have the cytochromes, and as you know, cytochrome is going to play a very important role in the review article that we're all going to assemble. And so one of the major functions that cytochrome plays is that of electron transport and facilitating ATP synthesis. And I've alluded to this previously, that ATP is a molecule that essentially helps to store energy. Another really important iron-based protein, and here we're talking about an enzyme, ribonucleotide reductase. It is responsible for the formation of deoxyribonucleotides, which are the building blocks of DNA, so used for DNA replication and DNA repair. Alright, so we can continue on with this list, and we can see the variety of roles that the metal plays. What I want to show you is a video that was made with one of your classmates where we can see how we're able to obtain iron in our body. So food is a source of iron, and we're going to look at an example of cereal being fortified with iron and how we're able to extract and I will talk about how our body is able to mobilize that metal. So in that video, what Andrea basically did was she took cereal which has been fortified with iron, and if you looked at the label, it actually, one cereal provided about 70% of the daily intake of iron that we need. And so in that activity, she used water to homogenize the sample. Then she exploited the magnetic property of iron in order to be able to isolate the iron filings that were present in the cereal. So what is being used to fortify the cereal is actually the metallic form, the neutral form. All right. So that is one way that we can ingest iron. And as you can imagine, that material isn't how we will actually utilize the metal. So in our bodies, we have to metabolize it. We have to biotransform it in a way that it becomes bioavailable and useful. Other forms of iron can be iron in the 2+ form, let's say, for example, heme-based iron, or non-heme iron, iron 3 as an example. So these can come from red meats. So we have to think about this idea of orally ingesting iron, and then that iron having to go through the environment of the stomach. So the stomach is acidic, about pH 3 to 4, and then in the process of metabolism, the acidity is even higher, so we have a lower pH. So let's think about what happens, or what is the biological fate of the metal as a result of digestion. So essentially, your body has to convert the iron into iron 2 if it's not already iron 2. And even if it is iron 2, it needs to be able to extract that metal from whatever molecule is coordinating it. So in the example of the fortified cereal, where we were talking about elemental iron, and it is dissolved via acidification, coupled with oxidation, generating iron 2+. So here is just the chemical reaction where acidification allows for the iron to undergo a 2-electron oxidation, and the proton is what actually is getting reduced. Then we also have dietary iron 3+, which has to get reduced to iron 2. And so this is a process that can be facilitated by proteins or enzymes that are reductases. So these can be found on the cell surface. You also have small molecules that can play the role of reducing agents in the diet, for instance, ascorbic acid. When the iron 2 is available, it is bound by gastroferrin and it is taken to the intestine. There is an iron 2+ uptake system that shuttles the iron from the intestinal lumen into the enterocyte. So these are the intestinal cells lining the small intestine via the divalent metal transporter, abbreviated as DMT1. So here, this particular transporter has the properties that we've talked about corresponding to secondary active transport. So this is an iron 2+ proton symporter. It uses a proton concentration gradient and the electrical potential of the membrane to drive iron 2+ uptake. So here I provide a visual of the key things that I've just discussed. This idea of iron 2+ or even iron 3+ in heme being bound by a transporter in the receptor. So here we see what is essentially an endocytosis process. That metal has to be released from that particular molecule to then have the iron 2+ that's then shuttled into the enterocyte. Other ways of uptaking iron can come from, as I mentioned, the reduction of iron 3+ to iron 2+, and then the shuttling in of that iron 2+, the ion itself, via the divalent metal transporter, and that is through this symporter with a proton. All right, so then we have the iron within the cellular environment. Part of that iron is going to get stored in the protein ferritin, which basically produces iron nanoparticles. The other part of that iron is what the rest of the body will depend on. That iron gets shuttled to the membrane, and then we're going to talk about this process of oxidizing that metal from iron 2+ to iron 3+. So iron 2 plus is exported across the enterocyte membrane into the bloodstream by ferroportin, and this process is regulated by the IREG1 protein. There is a critical oxidation event that takes place before the release into the bloodstream. And so we have this protein, this heptastin, which will oxidize iron 2 to iron 3. This is a copper-dependent ferroxidase. Other cells employ ceruloplasmin, which is another important ferroxidase. It is a copper-dependent ferroxidase. And another feature of this protein is that it is a major copper transport protein. All right, so this protein actually then plays two really important functions in terms of regulating iron, facilitating its oxidation, but also regulating copper biodistribution. All right, so then we see that event here, this heptastin, which will facilitate the oxidation of the iron. And then what happens immediately when that iron is about to be released is that there's a protein called transferrin, which will bind two ions of iron 3, and that's going to help to facilitate the transport in the bloodstream. All right, so let's talk about this, why that process is important. And so this relates to iron 3 plus solubility. Absorption from the intestine and into the blood, so we're talking about a pH of 7.4, right? So we've moved away from the acidic environment of the stomach now into the basically neutral, slightly basic environment of the blood. And this results in oxidation. So now iron 2 plus is iron 3 plus. Interestingly, at this pH, precipitation would occur. So what forms is this iron hydroxide compound, which is irreversibly insoluble, excuse me. But there's going to be a small amount that is soluble. Okay, so let's try to get a gauge as to how much. All right, so we think about the solubility of this particular iron hydroxide. Okay, so this species. So this the dissolution will lead to the formation of the iron 3 plus ion and 3 equivalents of hydroxide. So we need the solubility product, which is a measure of how much that dissolution occurs, that dissolution coupled with ionization. And that equals to the concentration of the iron 3 plus times the concentration of the hydroxide raised to the third power. So this KSP value is approximately 10 to negative 38 molar. Now, if we want to determine what this corresponds to in terms of the concentration of iron, basically the soluble iron, well then, at a pH of 7, we have to use this equation here, convert the hydroxide into a concentration. So we have the pH was a measure of protons, but it can also be a measure of the hydroxide concentration. Plug that into this equation, and what we obtain is the concentration of iron 3 plus, which would be 10 to negative 17 molar. This concentration is extremely low. Basically, that iron would be biounavailable. So then, the fundamental question, why oxidize iron 2 plus when it is far more soluble than iron 3 plus at a pH of 7.4? Let's explore this mystery. So, there are possible fates that iron can undergo in the body, released into the bloodstream. So iron 2 plus can generate reactive oxygen species via Fenton chemistry. Now, reactive oxygen species are extremely important. We thrive on them, ourselves need them to facilitate the formation of a number of molecules through the interaction with these species. The problem is when the generation of these species is uncontrolled, and then that can cause great harm to the body because it can lead to cellular damage as a consequence of redox, uncontrolled redox, and so you lose that ROS homeostasis, and this can trigger what is known as oxidative stress and apoptotic death of cells. So apoptosis is a programmed cell death, right? But you don't want this to be something that gets triggered in an uncontrolled manner. Iron 3 plus, therefore, when it's generated, has to be regulated completely at the level of uptake. Because if it's not, what you'll get is that precipitation event forming iron hydroxide, and again, virtually irreversible, so then that iron would no longer be functional or available, or it can very easily be reduced by reducing agents within the blood environment. There are a significant concentration of them in the bloodstream, so that iron gets reduced from iron 3 plus to iron 2 plus, and then you have your uncontrolled reactive oxygen species. But in fact, our body has developed a molecular mechanism to control the iron that is released into the bloodstream and to facilitate its increased solubility and its delivery into cells and biodistribution. That is the serum transferrin protein, which can bind two iron 3 ions. There is some speculation of low molecular weight molecules facilitating the binding of iron and its potential, perhaps, uptake from when the iron 3 plus is generated and released into the blood, and then delivery to serum transferrin. But generally, what's understood about the pool of iron that is bound to low molecular weight species is that is a consequence of a possible disease condition where there's too much iron and you saturate the serum transferrin protein, and now you have iron that's freely available, that's labile, that gets bound by this low molecular weight species, but that speciation could then very much lead to reduction of the metal and then again facilitation of reactive oxygen species. So the contribution of the low molecular weight pool is not thought to be significant. It is believed that serum transferrin will virtually bind 100% of the iron and help to then transport it throughout the body.